Professor Ricky Johnson, take one.
Thank you. Good morning.
Good morning.
How are you?
Great. I'm fantastic. Great to see you.
Might as well start with science.
Hmm.
Why and how did you get involved in science?
It wasn't an active decision. It never was. I was talking to someone the other day, and I reckon I was around in year 8 or 9, and I told my mum, I think I'm going to be an electrician. Don't know why, but I just thought, yeah, that'd be a pretty cool job. And as my high school education continued on, I thought, what do I really like? And I always liked chemistry and biology, not so much math. And I just followed my path. I went to a local high school, lived in a very blue-collar part of Melbourne, and science wasn't a strong suit for the high school I went to. But I had some great teachers who encouraged me and helped, and just fell into it. And I kept enjoying it, and to this day I still enjoy the discovery of science. That's the thing, right? You learn every day. You learn something new, and when you're in high school, you're learning out of textbooks, and now we're learning on the go in real time through our own experiments and our own thoughts.
So you left high school and went to university?
Yeah.
You went straight to medicine?
No, I did a science degree at Melbourne University, which for me was a very interesting experience. So I lived in Preston. It's a very working class, blue collar part of Melbourne. I don't think I'd been south of the Yarra River too many times in my life by the time I was 17, 18 going to uni, and got to university, and this whole world opened up to me of people from all different walks of life, backgrounds, and then the opportunity to just study and be in an environment that was just completely liberating. I loved it. Melbourne University was just an eye opener for me. Socially, emotionally, spiritually, and obviously from an education perspective, just best years of my life.
Exciting too, huh?
Yeah, I loved it. And we'd stay on campus, So first year I had 30 contact hours, right, a week. So you're there all the time. But in between lectures, you'd go to the cafe and have coffee, or you'd go to the pub and have beers. How good is that? And I was with people from other parts of Melbourne, indeed other parts of the country, who were coming in. Like, this whole world opened up to me. And at the same time, doing the thing that I really, from a education perspective, loved. I was learning about science. It's fantastic.
Was there an inspiration that came along during that time that pushed you towards research?
No.
What was your next step?
Yeah, so I did. I did a classic undergrad degree, 3 years, majored in pathology and immunology, and just followed my nose with that. There wasn't any. I never had these active decisions that I was going to do this, and I never had my world mapped out like some people do, and I always sort of marvel at the people who can do that. I just followed my nose. And then I did honours. So honours is after sort of your undergraduate degree, you do a year in the lab. And so the first real, I think, entry point to research, because it's unstructured. You're doing experiments, you're writing a thesis at the end. And I really loved it. And it was also at the University of Melbourne. And for the first time, I think I had unstructured learning. And it just resonated with me. But at the time I thought, is this really what I want to do? So I took a year off and did what a lot of people did at that time, put a backpack on my back, went to Europe, went to the US, worked on a summer camp for 9 weeks. So I had some responsibility looking after 12-year-old kids when I was only 22 myself. And then I remember being at that summer camp in North Carolina, and it was coming to the end of it. I thought, what am I going to do? I want to get back to Australia. And I had this sort of standing offer from the professor at the time to come back and do a PhD. And I remember sitting around, it's this classic summer camp with a lake and, the quintessential American summer camp. I remember sitting around the lake thinking, yeah, maybe I'll do this PhD thing. I'll give that a try. So I came back and started the PhD, and it went from there.
In what area was the PhD?
Yeah, so it was in cancer research. So from that point, I got into cancer, and that's what I'd studied. That's what I'd studied my honours year on. So I always had an interest in cancer, not for any reason. Like I didn't have a particular drive. I didn't have a family member who died of cancer. I just found the biology really interesting. Right? I mean, if you strip away the emotion of it, it's a fascinating disease. Right? It's normal cells that go completely out of control and evade everything that your body and evolution has set up for us to survive. It evades that. So there's a sort of perverse beauty to it. And I know that sounds weird, but from a biology perspective, it's so fascinating and so beautiful with the way it all works. And of course, what you want to do is unpick it, right? I mean, once you're in there, you want to understand it. So that was the driver. That's the little drug there, you know.
Okay, so you kicked off and you PhD in cancer.
Yep.
Where to from then? How did that come in?
Yeah. So 3-year PhD, which was great and had a great time. I always had to. So my best mates are not scientists. They're my high school mates. I mean, I still now. Tomorrow night I'm going out with my high school mates still, right? And they all got into, there's some tradies and guys who got into banking and, they're just different. And I always had to explain my life to them and, what I do. And they always thought it was a bit weird and funny. And so I remember finishing my PhD and they're like, okay, so now you got to get a job. I'm like, well, no, this continues. And so the classic career path is that you go off and you do a postdoctoral fellowship overseas. And, it's a good way to get out and see the world. So I chose to go to Boston, which from a scientific perspective is one of the best cities in the world. And that had a bearing on why I went to Boston. But the other big part is it's got the 4 major sports teams in America, right? So I'm a big American sports.
What are the 4 clubs?
Yeah, so baseball, American football, basketball, and hockey. And Boston's got them all, and they're really, Boston's a big sports town. So, I wanted to go to a place that, yes, it was going to be great educationally, and yes, I was going to learn a lot, and career-wise, of course, it's going to help. But I wanted to enjoy myself. I wanted to be embedded in a place where I was going to have some fun. So Boston ticked all the boxes, and I got into Harvard Medical School to do a postdoc, which for me, yeah, okay, it's Harvard and I understand all the history of it and everything. But to me, it was a place where I was going to go do my postdoc, and that was great. And this was back in the 1990s, early 1990s, so pre-internet, right? So you wrote letters to get jobs and things and faxes. And I remember the letter, I was still living at home at the time, and a letter came to home, and Mum said, oh, there's this letter here, and it's got this red seal on the back. Harvard was very much into the, it wasn't quite wax, but it was sealed letter. And it was a letter of offer. And I opened it up, and I'm like, oh, I've got a job. Great. And Mum burst into tears. She's like, what, you're going to Harvard? I'm like, well, yeah. And I didn't really think much of it. But for Mum, who's, she was a teenager in World War II and came through. I mean, she was telling me about, living in an era where there were rations and ration cards and all that sort of stuff, right? So, I don't know. I think it hit me then that what I'm doing, I shouldn't be so, not blasé about, but there's another level to it, and I needed to actually accept that and embrace that. It came through my mum.
I just think you're being Australian about it.
Yeah. And it's sort of, it's a, it does take some of the pressure away when that's your mindset. That's the way you're inherently built. But it did probably hit me now that, okay, there's a bit of seriousness here that needs to come with it. And it probably for that first time, it really hit me. And then I got to Harvard, and then when it really hit me, because it's a fascinating place. So you basically walk in and you're surrounded by everything that you could possibly hope for and wish for to do your science, to answer the questions that are in your head. It's a very challenging environment though, because they say, here it all is. And if you fail, there's only two reasons why you'll fail. One, you're not smart enough, and I'll actually tell you that because you're surrounded by all these brilliant people. I mean, I went to lunch on like 3 weeks in, and there's 2 Nobel Prize winners sitting over there in the cafeteria. That's, you know, the environment. So you'll fail if you're not bright enough or you don't work hard enough. And so one of those things you can control, so you work your ass off, and the other is in the lap of the gods, you know, whether you've got it enough.
Did you find it daunting? Or?
A little bit, yeah, because, you know, big fishes in small ponds in Australia, and then, you know, you get over to the US and you know, it was like, well, the lab that I joined was like the United Nations. There were people from all over the place coming there, and they'd all come there for a reason because, you know, it was the best place to come and do medical research, and they wanted to build their careers. So it was pretty competitive. It was very competitive, actually.
What happens when you said the lab I joined? So tell us the process of joining a lab.
Yeah.
Whose lab? Because I know most people won't understand how that system works.
Yeah. So at the time, again, so, you know, I finished my PhD in 1993. So at the time, there were positions made available that were published in scientific journals, you know. So a laboratory at Harvard would have some funding available, so they put a job ad out and you'd basically apply for it, right? And so you chose the lab based on who the leader of the lab was, you know, what sort of reputation they might have or what they might want to bring to you in your career, and then the direction of the science. So I chose a lab based on the direction of the science mainly. I wanted to work in a particular field, and the head of that lab was actually a young guy. It was he was just starting off, and I thought I'd rather do that and be part of a sort of exciting new environment than a person who was very well established and basically had this sort of factory and you'd just be a little cog in the wheel. I wanted to be part of something that was growing. So they were the drivers for me joining that particular lab, and it worked out really well because I learned a lot from him as he was learning. Right, we're all learning on the go. It was sort of chaos.
Sounds exciting.
It was brilliant, you know, but hugely competitive. So at Harvard, you know, you got 11 years to make tenure, right? So the clock's ticking. So tenure's the pinnacle for lots of people. You know, you become a tenured professor, job for life. You know, you've, in many cases, or many thoughts are, you've sort of made it. But all these new lab heads were starting at the same time. They'd had this influx of new people. So everything was highly competitive, which made it exciting, but also, as you said, a little daunting, actually, yeah.
So it's like a bottleneck. in
Yeah.
people.
Yeah, and 5% of the new people make it to tenure after 11 years. So everyone's going pretty hard.
What came out of Harvard, apart from you at the end?
I learnt to be very focused. That's when I spoke about going from being maybe a little blasé, probably a little naive about, where this was going to take me, to being a lot more focused, a lot more driven, actually. Yeah, and I realised this is what I wanted to do. So I realised at that point, I want to be a researcher for my career, and I really do want to make a difference. And I realised then you can have a bit of an ego about this, and as long as you don't get too carried away, it's not a bad thing. So, you take care and pride in what you do, and part of your life you'll be known for what you do and how you conduct yourself. I learnt I saw some bad behaviours there. I won't, some people who were sort of arseholes, to be honest. The way they went about it. It was dog eat dog, and it was all about the victory at the end and pushing people down. And
Apparently you had a lot to say about that.
yeah, and I didn't want to be like that. I think you can still be a good person with good values and do great science, and your legacy at the end will be more than your CV and how many publications you've got. Someone once said to me, they don't put your CV on your tombstone, and it's so true, right? So, but you can do all that. They're not mutually exclusive. You can be an excellent scientist, you can be a good person at the same time, and have a bit of fun,
So how and why did Ricky come home?
So, look, while I loved America, it's not the place I wanted to grow up in. So this was, you know, see pre what's been happening recently. So, you know, the Clinton era, and it was still a pretty good place, but there were parts of that society that I didn't like, you know, obviously the guns and that part of it. Socially, it didn't quite resonate with me. I mean, the disparity between the hugely privileged and the underprivileged is massive. So that didn't resonate. And I got a bit homesick, you know, I just like, you know, my mates and my friends and my friendship group are everything to me, and I sort of miss them. And my partner at the time, and we got married when we came home, we just thought this is not where we want to be and where we want to live, so we came home.
Wow. What did you do?
Yeah, so
I did the same thing from the UK. I came back home.
yeah,
Now what?
so I came back, and about 2 years after I came back, I came to Peter Mac. So that was 2000, year 2000, and it was my first independent job. So I went from being a postdoctoral fellow, where you're working in a, you know, as part of a team, but you've got a boss, a lab head, to now running your own lab. So independent, and that was my first independent lab was here at Peter Mac, and I'm still here 25 years later. So must be something about the place that I love, which I honestly do. And again, that was another moment of awakening and clarity, because there's responsibilities. So now you've got PhD students that you're responsible for. So you're responsible for their careers and sort of their lives, actually. And then you're employing people. And so, well, you're responsible for them being able to pay their bills. And, you know, the way that it works, you've got to write grants. And so it's not just for your own salary, it's for your team. And I've played team sports all my life, a lot of footy, and I think that grounded me in how you do work as a collective, without it being too cliched. And that was another sort of level of responsibility that started to ratchet up too, because it wasn't just me now, it was, you know, the group, and how they were going to flourish with their careers, but also how they were going to take their lives forward.
Now your journey with cancer.
Mm.
Your journey. You decided you wanted to do cancer research. Where did you start, and where did it lead you?
Yeah.
I know it's going to be a difficult question to answer.
No,
Or
but
maybe it's not.
look, so I'm very privileged in having my career happen at a time when technology has gone nuts. So there's always been questions that I've asked from a biological perspective. You know, how does a normal cell become a cancer cell? How does a cancer cell continue to live and proliferate and grow and expand? How do we combat that? You know, whether we intervene in some way, shape or form with a drug or something else, or how does the body try and combat it? And it's this internal fight that's going on for people who have cancer between the tumour, which is like a foreign body, and the normal body, which has its own mechanisms, like immune mechanisms and other ways to get rid of it. So they're the big questions that I had, but we didn't have the technology to answer those questions previously. But then when I started, we're in the genomic era, right? So the first human genome was sequenced, you know, in the 2000s when my career was taken off.
Could you explain the genome?
Yeah, so it's basically, you know, the genome is our DNA, which makes us all up, and we're all inherently different with our DNA pattern, you know, which makes us individuals, but we've got common human, you know, a common human genome. And it took, 13 years and $3 billion to sequence the first human genome. And now we can do it in a week for $1,000, right? So you just think about over that 10 or 15 year period, how technology has driven that advancement. But having the first roadmap, if you like, for what makes humans human was revolutionary. And that happened, you know, coincidentally, luckily, when I was, you know, my career was really coming together. So you can use all that information. So the technology was going crazy. I mean, just think about, you know, late 80s or something, what a mobile phone looked like. I think it was a big brick, right? You carried it around like this. You know, it was ridiculous. And then, you know, not too long later in the 1990s, the Nokia came out, and it was revolutionary. It's this little phone, and, you know, you could send these little messages and funny little, what look now we'd call them emojis, but little ways to pattern a smiley face. And then of course smartphones came on. So think about that, and then overlay that into science, and that's what was happening, right? And so it was just going, and so it was incredibly exciting because you had these thoughts in your head and these ideas and these questions that you were just busting to answer. And now for the first time, the technology was allowing us to answer it. So understanding the genetic basis of cancer, right? What goes wrong to our genomes that drive the transformation of a normal cell to a cancer cell? For the first time, understanding that and seeing that there were some commonalities. There were some genes that were often mutated in cancers. And so they were genes that we needed to study and understand. And the area of science that I got into is called epigenetics. So it's the way that genes are turned on. So we've got roughly 20,000 genes that make us humans, but they all need to be turned on or off in the right way to make a hair cell versus a skin cell versus a muscle cell, because it's the genes that define how the trajectory of a cell and what it's going to be and the function of that cell. So that's regulated at the epigenome level. It's the way that our genes are turned on or off, and it's really around the way that the DNA is packaged within a nucleus. So it was fascinating, and all this was coming about during the 2000s when my career was taking off. So we had technology, we had these breakthroughs in biology, and it was just the most exciting, and still is the most exciting time to do science, in my opinion.
What cancers did you target?
So
or what appealed to you? Yeah, because it's a broad area.
yeah, it
And
is.
you decided you've got to make a choice, I guess. Or do you?
I did, and I decided to work on liquid cancers, so blood cancers, so leukemia, lymphoma, multiple myeloma. And the reason was I wanted to study it in not just in the human context, but with mouse models. So mice can be a fantastic model for cancer, and at the time there were some brilliant models of leukemia and lymphoma that had been developed. These were genetically engineered mice, so these mice had their genomes changed so that they then got a lymphoma or a leukemia that we could study and we understood it, and we could then look to see how we might intervene. So could we develop new drugs or use existing drugs to try and intervene in some way, shape or form in this mouse model, and then take it into the human setting. So this way of translating research from a laboratory setting into a human setting. So I was interested in the blood cancers, and I still am. That's my major focus, and mainly was because we had these so-called tractable models, these models that we understood, that were very well defined and that mirrored what happened in the human context. So the genetic changes that happened in the mice that we engineered into the mice are the same genetic changes that occur in humans. So it was a really nice model system.
And you did solve some of it.
Yeah, I mean, I'd like to think we have, so I spoke about epigenetics and how we turn genes on and off. So as it turns out, and probably as you might expect, that happens in cancers as well. So there's certain genes that are turned on when they shouldn't be, and turned off when they shouldn't be, and that can be an epigenetic underpinning or driver of cancer. So fortunately, again, serendipity and just chance and being in the right place at the right time, there are now drugs being developed that actually could alter that. So the genes that were turned on in cancers that shouldn't be, now we could add a drug and turn them off. Well, that's pretty cool. And we could now apply that to our mouse models and then into humans. So I was involved in some of the very early clinical trials. I'm not a clinician, but I had clinicians here at Peter Mac. Miles Prince was a key one. He's a hematologist, and we were working on these new drugs that were targeting this epigenetic process. And we went from mouse into human, and I got to see firsthand some of the effects, and some of them were breathtaking, so, just amazing, and that gave me real sense of purpose for what I was doing, and it was great.
Must be amazing feeling.
Yeah, it was actually, and and it's the thing you can explain to someone. So you can sit in a taxi, you're in a cab, and Cabbie says, well, what do you do? And you try and explain to him about epigenetics, and it's a hard one. But you say to someone, I work on cancer, we're trying to develop new drugs. We test them in our mice and people understand that. And then we translate that and then we try them in early phase clinical trials at Peter Mac. And we have patients who respond and immediately they get that. Oh, okay, that sounds pretty cool. And, it's a nice way to communicate what you do without dumbing it down to the point where, people are intelligent. They'll get it. You've just got to explain it in the right way.
I so agree. One of the questions was going to be, what do you love about your job? But I think you've answered it, unless you want to have another crack at it.
It's the discovery, right? It's that moment when you've got an experiment in a lab and you get a result and you're like, holy shit, and so often it can come like not what you might expect. It can be this other finding that you've got that completely comes out of left field. That's the drug, right? I mean, that's the little addictive thing that keeps you coming back.
And for anyone,
yeah,
it's this big picture. It's you're a child of the 70s, space program.
yeah, all that.
I mean, research in itself. Can you fail in research, or is every discovery, even if it's not a discovery, but is it a setback? But you discover something that's.
I think knowledge creation is underappreciated in our society, right? I mean, we're too hung up on, well, you've got to have something that leads to some sort of translation outcome. Either you're going to make something that's going to make $1 billion, or you're going to cure cancer or cure diabetes or something. But along the way, there's all the little bits of knowledge that come into play. And that sort of fundamental knowledge creation is something I don't think we appreciate well enough in society. So every little bit of knowledge counts. And it's all a little piece of the puzzle. And often the trick is to pull it all together.
The history of research itself, are you familiar with much of it? Like going back before your time, or
Little bits. I mean,
and has anything inspired you from?
It's always about the people who completely apply themselves to their craft, right? And don't accept the naysayers. And there's lots of examples out there where people have been poo-pooed for their idea. So let me give you an idea of how that came about. So we're in the era now where the traditional ways that you treat cancer, either you cut it out with surgery, you burn it out with radiation therapy, or you kill it, you poison it with chemotherapy. But now we're in the immunotherapy era, right, where we're harnessing the immune system. And this is something that I've studied over my career as well. And in fact, I was just in a seminar, I said downstairs, and it was all about immunotherapy and ovarian cancer and how, we're changing that. So, but there was a time in my career when I was working when people used to think that this is all rubbish, that the host immune system actually doesn't fight cancer. It's there to fight bacteria and parasites and viruses, but it doesn't have much to do with cancer. And there are a lot of naysayers out there in the scientific field who didn't believe it. And so, not me so much, but my colleagues, I saw them have to go through that and jump through those hoops and continue to work in an area where there was a lot of skepticism. And they had to hold their nerve and they had to trust their own data, right? So that's everything. That's the purest part of our job, is the data, because it never lies, right? It is what it is. And so I really admire people who have stuck to their guns in the face of, a lot of skepticism, a lot of people, second guessing what they do. And in the end, immunotherapy's come through. It's, revolutionised the way we treat cancer at the moment, including here at Peter Mac.
Look, Ricky, it's so exciting. I think it's exciting. I'm loving everything I'm learning about. That was good timing. Yes. Then I'll ask you a few questions in a bit. But before we do that, I lost my train of thought. No.
Welcome to Peter Mac and the list.
The Australian Cancer Research Foundation.
Yeah.
How's it assisted you?
So ACRF.
No, could you say the Australian Cancer?
Sure. So the Australian Cancer Research Foundation, I think it fills a void that nobody else and no other organisation in the country fills. Right? So it has a very strong sense of purpose, which is to provide infrastructure, equipment for cancer researchers and organisations across the country. No other funding agency in the country does that. And so, you know, the Australian Cancer Research Foundation have been doing this for more than 20 years. I don't know, is it $200 million or more that's been pumped into the system? That's extraordinary. And as I said, right, we've got this technology revolution that's driving us to, you know, answer the questions that we could only think to answer before. So you've got to be able to do that. So how do you do it? Well, you've got to buy the kit. So how do you buy the kit? Well, you go to the ACRF and you write a great grant and they support it and they chuck the money at you and then they challenge you to really deliver on it. And it's great. I mean, I love the philosophy, right? We're going to give you the opportunity. How awesome is that? You know, isn't that what we all want? You just want a chance. And then you'll have crack.
And someone to trust you.
Yeah, and to hold you to account too. And that's okay. You know, we provide reports back to the Australian Cancer Research Foundation. We interact with the board, we interact with management there, we interact with our peers through the, you know, Medical Research Advisory Committee. So there's all the checks and balances, but that's okay. You just want a chance to have a go at it.
I'm going to pass you over to Ben now. Ben, you...
Okay.
Would you like to?
Sounds great.
Yeah.
It's been a pleasure.
Yeah,
I'll
that was
see you at the
great.
end of this.
Yeah, no worries. Hopefully that was
Oh, keep
okay.
rolling. It was more than okay. You're great. Thank you very much.
Thanks.
I get to sit down.
Yeah, that's right. The seat's warm and everything.
Okay, well, I was thinking of trying to frame everything through the prism of sport,
Mm.
because you said you went to Boston because fundamentally they had four of the top sports teams, and we were here last year for when Sandro was given an award,
Mm-hmm.
and he talked about cancer research being a team sport.
Yeah.
And it was a really interesting thing. And you run a team of 750 people here. So I was saying to Tom Derrick, you're kind of Alex Ferguson. You may not be kicking the ball around, but
Yeah.
you obviously can. And could you sort of talk about things through the analogy of sport?
Yeah, science and medical research is a lot like sports. So it is a team game, and you have to have everyone, I guess, working in the same direction to get the right outcome. But you've always got your superstars, right? In any team, you've got your players who are, you know, they're just wizards at what they do. Their craft is something that only a certain few can achieve. And so you've got to harness that and support that. But also you've got the players who turn up every week and do the job that they have to do, and it may be not the most glamorous of jobs within the team, but it's so important to allow the superstar to do what they do. And so I think it's exactly the same way in a laboratory, or even at a broader sense across Peter Mac and the whole research portfolio. And you've got to celebrate the person who's doing the hard yards, the sort of the dirty work down the back, just as much as you celebrate the person who's scoring all the goals, or, you know, being the messiah who's delivering the ultimate win for the team. So, and that's a trick for management, right? Not a trick, it's a challenge. And I find that always a challenge, to make sure that we are celebrating everyone that's part of the team, if you like, but also giving people their appropriate rewards and dues and recognition. But keeping it together is always a challenge, you know, and there's external pressures that come into play. People want to pinch your stars, you know, it's like you know, the EPL, right? Where, you know, you got a star and Manchester City wants that player, so you got to try and keep them, but you can't give away the farm to keep them. So it's the same sort of thing. You are managing, you know, a very complex environment that's highly competitive. Actually, science is, people don't understand, it's massively competitive. It's driven mainly by ego, scientific ego, which is great because it means that people are pushing very hard. But it also brings out some character traits that you might expect, you know, in that ego environment.
You did mention ourselves before.
Yeah.
Hailing Big Two in Boston. So
Yeah.
that's sort of interesting, that. But what, if I understand it right, ID3, which paper you recently,
Mm-hmm.
is that, could you say that's like having your immune response bench player. If you carried that.
Yeah, look, you know, if you
Well,
like.
Can you explain it? Because this is the latest publication. Is it?
Yeah, we had a paper that just came out, and actually the best analogy for that was team science. So we were working on a part of that story, and it was actually about how these T cells may be educated, or in some cases re-educated, by some of the environmental cues. So there's these molecules called cytokines that come in and bind to cells and drive them to do a certain thing. So we were working on that. At the same time, colleagues across the road at the Darwin Institute were working on genes like ID3, but also looking at the same sort of process, how T cells may be educated, how they may become the sort of T cell that may actually have an anti-cancer effect. So we're working over here, they're working over there, and we realised that in fact we've got the same sort of ideas, but we've got different pieces to the puzzle. And so you know what we did? We sat down and we said, okay, open forum. Let's play all our cards on the table. Let's just show a bit of show and tell, right? And let's be really open about this, about our story. And we put the stories together and we're like, okay, your story is going to be pretty good. Our story is going to be pretty good. We put it together, it's great. So let's just do it. So we decided then and then that we were going to write a joint paper. We were going to share the spoils. And so the spoils are really authorship and, you know, where you are. And we agreed to then finish the story up collectively together, and we got a beautiful story out of it. But as individual stories, they would have been good, but not great. So that's team science. Expertise across the road at the Doherty, expertise here, and people actually coming together to get best outcome.
Was that an off chance meeting on a bus or in a pub? Like, how did you two actually know what was going on?
Yeah, the environment here in Parkville helps because they're across the road to start with. How did it go? I think it came about because there was a postdoctoral fellow in my lab who knew some of the scientists at the Doherty, and they were talking at a seminar or a conference or something at one stage, and it was very clear that there was some overlap. So then we got together that way. And that's where these sort of scientific exchanges that can be, you know, within a very organised forum like a conference, or even at the pub, or, you know, catching up, seeing them in the street. Sometimes that happens. We bump into each other. Oh, what are you doing? What are you working on? Oh, that's interesting. So we're sort of in that area too. Or I know someone that's in that area. Why don't you go and have a chat to Josephine Bloggs, who's, you know, either here or at Weehai or somewhere else.
Because I find that fascinating. You think you hear about Watson and Crick going to the Eagle Hotel.
Yeah.
And DNA.
Yeah.
Well, Rosalind Franklin was doing all the work.
Yep.
There's like, should ACRF sponsor the tea room? There's so much interaction that kind of happens. Like what? Sorry, I'm going off on a tangent now. But so going back to that team, so you had an overview, you could see what was going on in two different
Yeah.
places, you put them together.
Yeah.
So that must be your daily life, seeing so many things that are happening here.
Yeah.
You're in a unique position to go, wow. Tell us more about that.
Yeah. So look, my job. So I'm the executive director of all research here at FEDAMAC. I've got my specific domains. I'm a laboratory scientist, but you know, I get to, I guess, drive direction and strategy for all the research. So you get this helicopter view of what's happening, and my view on that is, you do try and direct and you pull things together, and sometimes, you know, you have to use a carrot, and sometimes you have to use a stick. But you know, often you just need to push people in the right direction and let them do their job. But to have that overview and just to be fascinated by it all, that's a privilege. Every day I come to work, I mean, I spoke about discovery, so now I'm going from individual discoveries to global discoveries across the whole place. And sometimes you just sit back in awe of what's happening. And it may be a clinical trial, it may be a bit of health services research with someone saying the economic and societal benefit of doing this sort of procedure far outweighs, you know, anything we've ever done before. So let's reform, let's drive that reform piece, you know, at a national level. So it's all just elevated, right? But conceptually it's the same thing. It's discovery and just the thrill of it, right? Just at a different scale.
And you mentioned before that immunology was kind of like the wacky outliers.
In cancer, yeah.
Cancer.
Yeah.
Sorry, yeah, obviously. Immunotherapy. What's got wacky outliers status at the moment? Like, what are you, or another way of putting this is, what are your footy tips for your different areas of cancer research? Who's going to win the premiership? Is it immunotherapy? Is it glyconics? Is it like what? What's your footy tipping?
Yeah, I think, interestingly, one good thing that came out of COVID was this idea about RNA biology being important. So we've got BioNTech here in the building this week. So that's one of the companies that made the COVID vaccines. And they had this idea actually about this for years. And again, it was poo-pooed. You can't make a therapeutic out of a RNA molecule because you can't deliver it and everything else. And of course, they worked out the secret sauce to develop COVID vaccines. And look what that achieved, right? I mean, you know, one would argue millions upon millions of people saved through that technology.
What is a COVID vaccine?
Sorry, COVID. So,
No, COVID vaccine.
yeah, so
Say code.
COVID. So the part or the part of the COVID vaccine that really worked is that it's mRNA. It's RNA molecule, right? So it goes the way that our body works. We've got DNA. We make RNA, which is sort of like the message from off the blueprint, and then that RNA is the code to make protein. And so the COVID vaccine was taking a bit of the viral RNA and packaging it up in a way that our cells would then make antibodies against that and provide immunity. So the idea that we would have a vaccine out of RNA was something that I think there was a fair bit of skepticism about whether it would actually work, certainly at scale and in a human population setting. And BioNTech and other companies worked out a way that they could package that RNA so it'd be protected and could be delivered as a vaccine. So that very same technology now we're applying in the cancer setting. So as I said before, we know that certain genes are mutated in cancer. So if they're mutated genes, then they're not normal. So couldn't our bodies see that as being abnormal, like a virus, right? Or like a parasite or something else. So could we package up that abnormal RNA in the same way as we made viral RNA for the COVID vaccine and deliver that as a cancer vaccine? Remarkable, you know? I mean, conceptually, yep, is it going to work? We'll see. So I think that technology now that's come out of COVID and come out of a lot of work beforehand in fundamental, you know, sort of RNA technology and probably, you know, more around immunity and infectious diseases is now taking shape in the oncology setting. So if I had to predict, you know, huge breakthroughs, it's going to come through RNA technology. And the other is, you know, data science is going to rule because we're not just working with a, you know, data set that's this big, working with a data set that's that big, that from all different, you know, aspects, whether it's protein biology to RNA biology to DNA, and trying to then pull all that together. So analytics, you know, of course, AI, to pull all that information together to come up with, you know, new advances is going to take us a long way. So I think they're the two areas. The data scientist is going to be the ruler of, you know, medical research in the next decade.
One thing Dan talked about, one of the fascinating things about Peter McCullum is that you are doing from lab to bedside. I mean, you're in a unique position just to actually, and Kayleen was saying, almost doing things there, testing them with the clinician. Can you talk about that a bit?
Yeah, so we've got the full spectrum from fundamental science and discovery science and knowledge creation, and it may not be any immediate translational effect of that. It may not be a discovery that immediately goes into the clinic. So we celebrate that and we continue to invest in that area through to the translational pathway where we may have something that could be then tested in patients, then into clinical trials. So we do, you know, 200 new clinical trials every year at Peter Mac. And often it's the cool stuff. It's the first, you know, so-called first in human. So there's a new compound or a new agent and we're testing it for the first time in any human, sometimes first time in the world. So we've had, you know, world first in human trials done here. we then take that to the next stage of seeing, okay, can we implement that into standard practice? And then can we evaluate that? So what's the benefit to patients at a population level or at individual level? What's the healthcare benefits, you know? And we've got to think about, you know, the financial benefits of that. I mean, we live in a real world where, you know, we have to try and deliver things at scale, but to the benefit of patients, not just around their health, but also to the economy. And we recognise that, you know, we're playing in that game. And you can't just say, oh, the government's got to fund it. There's only so many dollars out there, and so we've got to find the most effective and efficient way to deliver that.
You talked about the first time in the world. So one thing that strikes us, having interviewed a lot of people for this, Australia seems to be doing a pretty amazing job in cancer research. Talk about Australia, like broadly Australia's role.
Yeah, so, I mean, look, you know, I could roll off a cliche, oh, we punch above our weight, and, you know, and that's, okay, that's a nice little soundbite perhaps. But I think there's a tradition here of medical research and science in this country that does resonate. And again, it gets back to sport. I mean, why is Australia, why does it punch above its weight in sports, right? It's something that I think is inherent to us, and I don't know why, but it is. And it's the same thing with medical research. I think maybe it's the challenge, and maybe, you know, it's a little bit of the chip on the shoulder, and, you know, we'll have a crack here. But I think as a society, we're set up to allow people to have a good crack, right? And if you think when you're a kid and what your parents want you to do, they just want you to have a go. They do, right? And as a parent, that's what you want your kids to do. So if that holds through and holds true, then it plays out in different ways. So it plays out in the sporting environment, and it plays out in medical research. So I think there are, the analogies can continue. And we have had, you know, massive successes here in Australia. You know, you think about some of the discoveries that have come through over time. You know, cochlear implants, as an example, completely revolutionised the way, you know, hearing impaired people live, their quality of life, as an example. And that came out of a discovery from here in Australia, here in Melbourne. So there's a history of that, there's a celebration of that, and I think there's the opportunity to have a crack.
So that you need. Sorry.
Hi, Ricky.
Oh,
Just wrap up. No, what's next?
Oh, what's next? Well, I thought we did that with the mRNA.
Ricky said more to say about it.
Okay.
yeah,
Do you
no.
want to build on what's okay? So what's next?
So, okay, well, what is next? I mean, I touched on data science, and I think, artificial intelligence is going to take us a long way, right? So I think, as I said, the computer scientist and being able to bring in this multidisciplinary teams, and we speak about that a lot, but it is mathematicians and engineers and, you know, computer scientists and biologists and clinicians all actually working in, you know, sort of on the same problem, if you like, is the way that we're going. And again, that's team science, but, you know, at big scale. So I think that's, you know, certainly where Peter Mac is heading and where this precinct is heading. And, you know, we here in Melbourne and other parts around Australia are starting to drive that. So the federal government is driving the development of comprehensive cancer centres. So Peter Mac is a comprehensive cancer centre now. It has everything, you know, within its four walls to be able to deliver that. And they're trying to set those up across the country. But then the next trick is, how do you link those up? So how do we set up a comprehensive cancer centre network? So we've been driving that, and, you know, I actually wrote a white paper on that went to government and has been adopted now through Cancer Australia to set up the Australian Comprehensive Cancer Network. So that will link up all the comprehensive cancer centres in the country so that we can share our knowledge, share our technology advances, share our people, so that the care that you're getting in Melbourne is the same as the care you'd get in Broome, is the same care as you'd get in Townsville through this sort of network. And so that's just bubbling along now, and it's coming to fruition. And I think that's going to hopefully be the next sort of revolutionary part of the Australian healthcare system in cancer at least. But it's all based on still the discovery science, still the fundamental, sort of idea that we need to actually pull in all these different teams because, it's a big question and we can't answer it just with one modality. It can't just be the biologists, it can't just be the clinicians, it can't just be the data scientists. It's got to be all of those things together.
I love the bit you said at the beginning about how fascinating cancer is.
Yeah,
Because it evades everything you throw at it.
yeah,
And it's kind of like you need to out cancer.
yeah.
So like can you ever be one step ahead?
Um,
I suppose vaccines.
Yeah, so that's one way. I mean, so the big area that I don't think we've invested enough in is prevention, and so there have been huge advances there over the years at a population level. So think about back in the day, Slip Slop Slap. Remember those campaigns for skin cancer? That changed the trajectory of skin cancer, and similarly for lung cancer with the anti-smoking campaign. So they're public health campaigns. But there's this idea about precision prevention, and that's at an individual level. So really, I think where we're heading is cancer being taken from a population level to a personalised cancer level, personalised cancer medicine. And so that's all wrapped up in this whole idea about comprehensive cancer centres and how we're going to deliver care that's going to be based on your individual cancer, the genetic basis and the epigenetic basis of your cancer, how we understand that and how we treat it. And if you take it one step further, maybe the predisposition to you getting cancer and then how we're going to treat you as an individual. So that's going to drive everything. And again, this is going to need big data sources and be able to pull that together and understand the interactions between, for example, genetic changes and the environment and how they interplay and collide to then give an ultimate outcome, which may be cancer. And we know that that happens, actually. So there's a beautiful study recently came out of the UK where people had genetic changes that by themselves were not enough to drive cancer in that patient. But if they were in an environment that had a certain level of pollution, environmental pollution, that caused an inflammatory reaction in that person that then drove the transformation of a normal cell to a cancer cell. Fascinating, right? Absolutely fascinating. So, but you need to have all the pieces of the puzzle together to pull it together. So all the environmental, climate information, and then all the epidemiology data, and then all the biology to come together to be able to pull that together.
So that goes back to your switches.
Yeah.
So your genes are turned on, and then an environmental thing.
Yep. But we've got to recognise that. And again, it's teams, right? You've got to have the climate scientists and the immunologists and those sort of people. And then you've got to have the biologists and the molecular guys and the data scientists. And there's not one person who can do any of that. So, you've got to leave your egos at the door, just like this paper we had, lay your cards on the table and then, come together and have a good crack.
I'm still intrigued as to how that exchange of information happens. Between, how do they interact? Because everyone's going to their own conferences. Where do they...
Yeah. There's something to be said for people talking to each other, surprisingly, and there's still a lot of serendipity that happens. You'll come across someone, or someone will know someone else. Oh, you should go and talk to, that person over there because they're doing this really interesting thing, or I saw something. So I'd like to say that it's hugely sophisticated and we've got all these networks and we map everything. That's not the way life works. There's a certain amount of serendipity. That's sort of beautiful in itself sometimes, when you think about it. It's sort of, I don't know, it's sort of, there's a sweetness to it when it's like that. And you can go back and you can think about how that story came about. And it may be someone who knew someone else who put you in contact, or you went and heard this talk, or you just bumped into someone. I don't know, there's a certain sort of romance about that I still love.
I'd like to know what if you've got a minute,
Yeah, sure.
what criteria do you use you select people to work here, right?
Yep.
It's your job.
Yep.
You're a fabulous team of people to work with. How do you do it? What's your golden rule for getting good? Because you do it very successfully.
We do, but like anything, and again, like teams, you go and buy a great striker from someone and they don't work out in that team. So, look, I always think that you get people in, it's like that, it's somewhat like that Harvard experience. You give them the opportunity, you try and provide them with the best environment, and then frankly, you get the hell out of their way and you let them do what they do, right? Because these are driven, smart, intelligent individuals who have their own sense of purpose, and the last thing you want to do is meddle with them. So I try not to meddle. I try and direct. I try and ensure that they're heading in the right direction. I try and ensure that they're being a good corporate citizen, a good part of the Peter Max society. But I get the hell out of their way and then wonder at what they've done, because often it's brilliant, And again, that's the joy of it.
Those are conditions where only the brilliant will survive. Because if you're just presumably, if you're letting them, basically they'll succeed or fail.
Yeah.
And it's more likely to get success if they're driven on everything.
Yeah, it is, and again, that's the competitive nature of the job, and you just have to accept that and embrace it, but do it in a way that you're not an asshole about it. You don't have to push someone down to get ahead. And I know that sounds a bit pithy and whatever, but that's the way. They're my sort of values, and I try and overlay those values on the rest of the faculty, and it works. I mean, we get good people here. The Sydney Swans had this no dickheads policy that they supposedly lived and died by. And, we're not overt about that. But we look at the character of people that we're bringing in, and that counts, actually. It does count. Their character traits. So that's part of the secret. Well, if there is a secret sauce, that's part of it. It has to be. It can't just be you're a brilliant person, but, you're a complete knob. We don't want you here. Go and do it somewhere else. Is that okay?
I'd love a job.
I get the best job in the world. I mean, I seriously do. The only frustrating part of my job is that, what I really love to do is the science.
Yes.
And I haven't done an experiment in, 10 years more.
That's the beauty. Talking to Kaylee, the most amazing thing is here you have a really smart person who's brilliant at actually designing the tests and the approach, rather than someone who, and far more practiced in it than someone would be in their own world. So just organisation, it feels like you're set up to succeed.
Yeah, I think organisation we are, but from a personal perspective, I'd still like to do the science every now and then. I don't know, if someone gave me a gift, it'd be like, yeah, put your lab coat on and go and work in the lab for a week, not go to meetings and do it. I still love that other stuff, don't get me wrong. I love the leadership stuff and I love the strategy and I love the direction. But, I still
So
love
if
to.
you're doing a PhD now,
Yeah.
what would you do?
In what area?
Yeah.
I would grab this mRNA area, but I would teach myself, informatics and computer science and be able to actually really drill down on data. Yeah, for sure. And I think that's the students now, the really good ones, they've got that, so they understand the power of computer science and data science, but they're good with their hands too. And they don't do experiments because you can. They do the hard stuff. Like they really try, very hard to have a crack at the really hard stuff. And because you can, the technology's there, so use it. Anyway,
Amazing.
Great.
I better run. Sorry, guys. Is that all right? That's brilliant.